So, friends, once again, welcome to the part two of my third lecture, Interaction of Water Molecule with Different Substrate. Now, what happened when these free radical, especially the hydroxyl radical, react with any unsaturated bond in the macromolecule, then it lead to a reaction called addition reaction. The hydroxyl radical itself attached to one of the carbon center, and then it lead to the generation of a radical center on the second carbon which is associated, which was involved in the unsaturation bond earlier. The another reaction is called abstraction, where the unsaturated bond between the carbon and the hydrogen got snapped away, and this hydrogen with its electron called hydrogen radicals react with this hydroxyl radical and form water molecules, and in that process it lead to the formation of a ion center in the carbon. So these are two reactions by which the hydroxyl radical react with the macromolecules. Similarly, with the solvated electron, it lead to the formation of ion radicals. So at one carbon, it get a negative charge, while the other it get a radical part. Similarly, the hydrogen radical undergo a addition reaction at the unsaturated bond, which lead to the formation of a ion center on one of the carbon atom. If the oxygen is present there and of already a ion center is there on the carbon atom, then this oxygen react with this carbon radical center and form a peroxyl radical. This peroxyl radical in itself is not very stable, so it lead to further reaction. And in case of a ion radical which is generated to the abstraction of the uh, hydrogen atom by a hydroxyl radical, then this again lead to the formation of hydroxyl radical. Again, the rate constant value suggests it is a second order reaction with a rate constant value of 2 into 10 power 9 per mole per second. So which means that it's a quite high values, and they are very, very reactive in nature. Uh, if they, we know that in the physiology, the many, transition metal ions are involved, and especially the iron role is very, very important in iron metabolism. So what happens if the peroxyl radical interact with iron in three oxygen state? Then it lead to formation of oxygen, and the iron itself reduced to the ferrous state. Then this ferrous state iron react with the hydrogen peroxide and lead to the formation of hydroxyl radical and a hydroxyl ion, and again ferrous state iron oxidized to ferric state iron. So the overall reaction is basically like the superoxide radical react with hydrogen peroxide lead to formation of a hydroxyl radical, hydrogen, hydroxyl ion, and an oxygen. So again, this is this reaction is called Haber-Weiss reaction, and it is very, very important because these are catalyzed by the transition metal ions, especially the iron is important reaction. So the Franklin reaction is also play a critical role in the generation of radical species. Although the the rate constant value as compared to the reactions which we have discussed in the previous slide is quite low, but the possibility is there for the generation of free radical due to a Franklin reaction. Now, how we will detect the free radical? Well, because the fundamentally what we are trying to ascertain that what type of species are formed in this radiation interacting reaction with matter, then and how to detect them and how to quantify that. This is our basic fundamental objective in this radiation chemistry. So detection is not possible by the conventional method, as we know that it's a very short lifetime of femtosecond, and if we go with the previous knowledge, the most lengthiest reaction is 1000 femtoseconds. Even though it is 1,000 femtosecond, it is not good enough to understand or investigate through a conventional method. So we require a more sophisticated and sensitive technique to understand on to quantify a qualitative detection of these critical, which is called pulse radiolysis. So here the production is recorded using a spectrophotometer which is connected to a cathode ray oscilloscope, and this cathode ray oscilloscope is tracing the and photographed the free radicals which form during the interaction of radiation with the matter or in the medium. Now, the Fricke dosimeter is another technique which is normally used by the radiation physics people to calibrate the radiation sources. So originally it's developed for the dose measuring device. Then the strong linear dependence of this application was that is most useful as a method to directly measure the number of species which are reactive species formed in the reaction. So what type of solution you require to form the standard solution of 1 millimolar ferrous sulfate in zero point 0.8 normal hydrogen peroxide, it's a sulfuric acid. Then when it is got irritated, when it's irritated by by a cobalt 60 source or some radiation source, then the ferrous oxidized to ferric. This ferric ion is generally generate a blue color that can be quantified using a conventional UV visual spectrophotometers. So that colorimetric response is linearity is very important, because unless it is not linear, that you cannot accurately measure the linear response and the response. So the dose response can be accurately measured up to 400 rays. So this is basically the sensitive range that the calorimetric dose response is lean up to the 400 gray. So if you from 40 gray to 400 gray, this process is very good to detect. Otherwise, if you go beyond the 400 gray, then you have other methods available which can be used to quantify the doses. Then oxidation is complete at 700 gray. So the most ideal condition is 40 gray to 400 gray. So beyond that 400 grays, you require another methods to quantify it. Now, what is the chemical reaction involved in Fricke dosimetry? Are very simple. The hydrogen radical react with oxygen, lead to the hydroxy peroxyl radical. Then hydroxy peroxyl radical react with the ferrous ion, and then ferrous ion undergo ferric ion and HO2 negative. Then HO2 negative react with the H positive and form hydrogen peroxide. Again, the hydroxy radical react with the ferrous ion, and ferrous ion again undergo a reaction to form ferric ion, and ferric ion with along with the formation of hydroxyl ion. The hydroxyl ion react again with the same fate as we have discussed earlier. Then hydrogen peroxide react with the ferrous ion and lead to formation of hydroxyl ion, ferric ion, and hydroxyl radicals. So the overall, it basically converts the ferrous to ferric, and the ferric convert to a blue color, and the blue color can be quantified for the quantification of the free radical, which is generated in this process. So this is a very useful way to measure the dose response. Now let's come to the second important part, the DNA. We know that DNA is made of sugar moiety, the nitrogenous part which is called bases, purine and pyrimidine, and these are basically held together in a form of a helical structure. Like these are two bases. They basically two strands which are stuck together to the different bases. Now, what happened during the process of any radiation exposure? These hydroxyl radical or any radicals interact with the DNA depending upon its linear energy transfer. If it is a high energy linear transfer, then the energy itself excite and ionize and create the radical center hydrogen. So there's no involvement of free radical. But in case of the low energy linear energy transfer, the hydroxyl radical or superoxide radical or any other soluble electrons, like these kind of species formed due to the radiolysis of water, and they further lead to the damage in the DNA structure. We know that the simple free radicals is of a very short life of 10 to the power minus 10 seconds. It means that 0.1 nanoseconds. And if you create a mathematical model, then only those is formed in the water column of 2 to 3 nanometer, maximum 4 nanometers. They are the one who can cause significant damage in the DNA structures. And that is why in my previous slide where I was representing the direct and indirect effect, I showed you a cylinder with a diameter of 4 nanometers. So it means that up to the 4 nanometers, the radicals who have been formed they are the one who can cause maximum damage to the DNA. So what damage they can do? They can create a lesion on the one or two, on the single or the both the DNA strands, or they can cause some basic lesions on the bases, and after that it undergo a number of reaction, and they can cause multiple damages. So let us go one by one. Now before going, you have heard about the concept antioxidant, because nowadays antioxidant is quite a very common name. Like, you name any skin beautification products and it contain antioxidant medicine, antioxidants, and it says you have a green tea and you will live for long and you will live like a young man. So what is antioxidant basically do that? So we know that for the inflammation, for the immune response, the free radicals are very, very important. So if anything is happening in our body, and so the first thing is that the free radical generated, and these free radical basically the signaling pathway for our immune response. So free radicals are basically essential. But if during any pathogenesis stage, or like for example, an external factor like radiation exposure, the concentration of radical generated is so enormous that they undermine the antioxidant machinery, intrinsic antioxidant machinery. So we have an intrinsic antioxidant machinery in the terms of some enzymatic enzymes, antioxidant enzymes like SOD and catalase. So they are not able to manage these the enormous population of radicals, and therefore there a state is created uh, where the balance is tilted in favor of radicals, and this is called the state called oxidative damage. This oxidative damage is lead to further processes like aging. They cause biological damages, and therefore the first more thing which any situations come to a scientist, that let's how to manage this oxidative damage. And that is the big thing is that the we have to supply the antioxidant in the form of, in a preventive way, or in the form of in therapy or something therapeutic purposes, so that we can able to correct this oxidative stress state, and that is the main function of it. Now, what these free radical species do, they cause either they cause single strand breaks, meaning that the single strand is involved in the damaged part, or if the concentration of radicals so high, or they may cause a double strand breaks, or maybe the single strand breaks may go to the double strand break. That is also a possibility. But if you talk about in a day on per cell basis, the single strand breaks is always high. They are something around 5 to 10 4. It means that something about 50 lakhs, sorry, 50,000 single strand breaks as compared to the 10 double strand breaks per day per cell. So single strand breaks, are quite high as compared to double strand breaks. Single This reactive oxygen species or ROS may cause base damage, because we know that there are two types of bases: nitrogen bases, pyrimidine and purine based bases are there, which may cause which play a significant role in the functioning of the DNA. And if this got hampered, then the overall function of the DNA will got compromised. So and beside that, our body have a refinery machinery which take players of all these base damage or single strand breaks or double strand breaks in through of nucleotide and base excision repair mechanism, or the single strand base repair mechanism. Then in the case of double strand breaks, there are two ways: by non-homologous and homologous recombination reaction, by which we can damage, which can repair these damage. But it depends upon the radiation dose. But if the radiation dose is enormously very, very high, then the repairing machinery got compromised, and therefore we required a medical attention to save the victim. Now, what type of oxidative damage we can expect in DNA in the case of radiation scenario? We can have a modified basis. We can have a basic sites aberration. It means the lesions in the bases. Then we have a single and double strand breaks. We can expect a DNA protein cross-linking. We know that DNA is surrounded by the protein, like many proteins which are critical for the role of the DNA. And if the radical center generated either in protein or in DNA, then the cross-linking of DNA and protein is quite evident. Then there are the aldehyde adducts to the amino base, basically to the anal formation is quite high in the case of multiple lesions. Now, what type of photons energy we can expect and what type of damage we can expect? So we know that when we move from visible to near UV, then the photon energy around 3 electron volt, and then in that process, the photosensitization is the only process. When the energy reach to far UV or vacuum UV, then the energy range values around 5 electron volts. Then the photoionization, or which may cause bases or 2-deoxyribose base to undergo ionization. But when we talk about in terms of soft X-ray or with a value of 25 kiloton volt, then the atom photoexcitation may take place with the Auger effect and lead to the damage in the DNA. But in case of gamma rays and X-rays with the electron of, in the case of X-rays which is 5 electron volts, or in a gamma rays with the mega electron volts, the DNA ionization is a significant process, and they all depend upon that the LAT values, that whether the direct effect will dominate or the indirect effect will dominate through the water radiolysis. But in case of any event which is more than the 4 mega electron volts, then the multiple effect, the cluster damage, maybe the pair production become the most dominant one and cause a multiple damages which are very difficult to analyze and make the picture further complicated. Now, this is what we have done. So I think we have uh, uh, learned about it through our previous slides. So I'm just skipping it for the sake of the saving time. Yeah, the time factor is very, very important. Like normally when the we scientists do experiment, then the time factor is very, very critical. If you somebody want to understand the effect of radiation in that femtosecond time scale, then the radiation physics or track simulation is the best techniques or experimental approach available to understand the excitation and ionization reaction. For example, Monte Carlo simulation. So you can see a radiation physicist people do Monte Carlo simulation to understand the effect of radiation in the this time frame. So once it is reached to around 100 to 1000 femtosecond, then I told you before, the pulse radiolysis is the best technique to photograph the generated radicals, and we can also quantify them with those photographs. But once it is reached to a time scale of microsecond, like 10 to the minus 6 seconds, then the events like target radical reduction or oxidation can be understood using the mixed techniques, something like pulse radiolysis and the conventional. But once as you move on with the longer time frames, like 1 second or 10 or 6 seconds, then there are the techniques called plug plating techniques or repair deficient cells or cell cultures become the most prominent one. But if I want to understand the long-term effect, how I will, what model I will like to employ, so then the animal model is the best one to understand whether the cell is getting proliferating or not, or whether there are lead tissue effects. Then in that case, the animal model is the best one. So according to your wishes and according to your objective, what you like to investigate, that you have to select the experimental approach. So in our case, mostly we focus on the cell culture and the animal model to understand the effect of radiation. So I was talking about the single lesion and multiple lesions, just like a few minutes of your, due to the single events such as a hydroxyl radical or ionization or secondary electron Auger effect, the single lesions are the most prominent one. But in case of the multiple lesions, the lesions take place in tandem, and this it may be due to a single event, or it may be due to the multi-ionization processes, which lead to several events and reactive species formation, and that can cause ionization of DNA, or they may cause water radiolysis of species, and maybe the involvement of secondary electron. So there are many processes which are simultaneously taking place. So in those processes, it becomes difficult to understand the damage part, and therefore the process becomes so complicated. Now in the oxidative DNA damage, if you see that They are oligonucleotide stand breaks where the hydrogen absorption can take place at 2 prime, 4 prime, and 5 prime carbon atom. Then there's a basic sites, which where the we know that the end glycosidic bonds are very very important for the different cell signaling pathways. So if they cause a hydrolysis of these end glycosidic bonds or they will get modified, then it cause the the compromisation of the function of the biomolecule. So that is called abasic site reactions. So they are like hydrolysis of N-glycosidic bonds or oxidation at C prime. They cause reduction in the innate reactions of the DNA. Then the base lesions, which maybe there are around 70 modifications have been already identified. I told you before, the bases are very, very important. So if any structural modification or uh, take place in these bases, then the overall function of the DNA got altered. Protein DNA cross-link, I've already told you before that because the DNA is surrounded with the histone protein. So if anything happen either in histones or in the DNA, then there are chances of protein DNA cross-linking. So they are the structures no longer available for the essential activity. Aldehyde adducts to the amino bases. This is very, very important. So it's the breakdown products of lipid peroxide. So lipids are also important biomolecules for the of the cellular machinery. So if the lipids are got breakdown due to the reaction with the anionic radiation, then the breakdown products of lipid peroxide and the oxygen products of 2-deoxyribo, they undergo and form the aldehyde adducts. So I will show in my next in my later on slides more clearly the mechanism of aldehyde adducts formation with amino base. Then there is alkyl labile site where the abasic sites and the few oxidized bases, including the thymine, glycose, and 5-fluoromyl urosyl hydrogen formation take place. So all those things I will show you in a typical case of let how the thymine will react with hydroxyl radical and superoxide radical. What type of model compound you are required to undertake these investigative studies? So you can take nucleosides and oligonucleotides. Normally we got it either we prepared by ourselves or we get it we use the services to construct the particular region, and then we understand and we conduct the studies. But the other thing which I personally like, because I'm a spectroscopist, which is like we have isolated DNA structures, like for example calf, the DNA from the calf thymus, or the plasmid DNA, which is of a bacterial origin. So these kind of DNAs are easy to handle. So whatever you feel like you generate a scenario, you call it damage in DNA in the intelligence and in the with and without the drug of your choice, and then you see that whether the DNA is undergoing any kind of damage or not. Then there are cell culture species or the tissue studies where the DNA in the can be isolated and can be performed with different technique, like comet assay we can perform, or other cell culture studies we can perform, and to understand the aspect of DNA or damage in the DNA. Okay, and let us see what happened. Okay, so we know that the base damage is the most prominent one, and there are 4 type of bases are there. One is thymine, which is almost completely available information in term of isolated DNA and model compounds. So thymine is one of the most studied DNA base as far as the radiation, ionizing radiation, oxidative damage is concerned. Then the cytosine, guanine again is a very very important, and they are the markers are available in the pathologies, where by studying the guanine oxidative products, we can understand the pathologies of different diseases. Then is the adenine, again is very apparent lack of information is available, and we need to be further checked whether what kind of investigation we can further. So this is a topic for the research for the expiring researchers. Now, with this, we can come to one of the very important reaction, thymine, and we can see that if the hydroxyl radicals react, and I have told you before, this is the unsaturated bond. If you can see my cursor, so one of the OH radical will go and attach one of the carbon center, and then the radical center unit on the other part. Now there are two possibilities are there. Which carbon atom the radical sorry, which carbon center will be generated. So if it that carbon center generated on this way, this would be a tertiary radical, or if the carbon center generated on this is a secondary radical. So we know that tertiary radical is more stable as compared to the secondary radical. So it means that the reaction will go dominate the tertiary part. Then this kind of radical centers generated. Another important aspect is the hydrogen abstraction. So hydrogen abstraction can be taken from this methyl group which is attached to the one of the carbon centers. So we can see that it leads to generation of CH2 radical in this part. The percentage wise this is very very less, it's only the 5%. Now the oxygen effect becomes the most significant one, but which I told you before, the oxygen effect. Now this oxygen undergo and react with radicals and form peroxyl radical. Peroxyl radicals. Now these peroxyl radicals undergo and form a variety of products. You can see that they are hydrogens, then they are the oxo compounds, then thymine glycols. So these are the products which are normally formed when the hydroxyl radical attack the double bond, on unsaturated site in the thymine. But what happens if the this CH2 radical center undergo react with oxygen, then this lead to the this a substituted acetic acid peroxyl radical. Then this undergo a reaction to form a, a alcohol and a formaldehyde. So thus lead to the formation of a important progress. So thymine glycol and the formyl uracil is the basically the two important products which are formed when the thymine base of the DNA undergo reaction with hydroxyl radical. Let us understand one more reaction which is very important. The guanine base when undergo reaction with the hydroxyl radical or the any oxidation reaction, then it lead to the center of ion radical center. Then subsequently, in the presence of oxygen, it lead to the formation of a just a minute. It lead to the formation of these hydrogens and the 8-oxo guanine formation. So this 8-oxo guanine formation is very very important because in case of any if you want to investigate the pathogenesis of any diseases, then 8-oxo guanine is a very important parameter, especially if you doing the kidney problems or liver problems. The doctors advise to understand the 8-oxo guanine level in the patient's blood or in the patient's blood samples, so they can understand that what level of pathogenesis has been all been achieved by the patient and the level of disease progression. So by analyzing this, we can understand the problem. This is a very important pathological parameter. Just a minute. Yeah. So if you I will not go into this much detail. So let us see that if we have a compare and comparative susceptibility of 8-oxo guanine and the guanine base with one electron oxidant in a photoexcited riboflavin basis, then we can see that 8-oxo guanidine response is very linear as compared to guanine oxide. So that is why the 8-oxo guanidine is a most important parameter as compared to guanine oxide when you talk about the basic oxidation of the guanine base. So this I have taken from a paper, a research paper from JOC. JOC is basically Journal of American Chemical Society, which is published in 2003 by the Revenat et al. So I'm just skipping the part because that's not much concerned with the with our audience. So I talk about it the tandem lesions. So tandem lesions that the OH part will got form a adduct with the amino bases. So this kind of adducts you can expect in these bases, and then you expect adducts formation, and because of adduct formation, what happens, the structural alteration take place, and because of structural alteration, the basic function of DNA got compromised. Similarly, this is a mechanism of formation of 8-oxo guanidine and amido tandem lesions, which I taken from the Docky et al. And it's published in Chemical Research Toxicology in 2002. And again, it's basically highlight the role of that how these hydroxyl radicals attack the 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 unsaturated part of the DNA base and then lead to the formation of a cyclic component. So I have that the four products very important. One is thymine glycol, then is we have a unicyl, then we have 8-oxo, 8-oxo adenine. Now, which one is the most important one? So if you see that and you compare the effect of intensity on quantum meter formation of oxidized nucleoside upon exposure of DNA to a UV lasers, then the quantum yield value is not much affected in thymine glycol and 8-oxo-guanidine. So this is very quite high. If you increase the intensity of up to 100, then almost whatever the damage or quantum yield we can achieve in the case of 8-oxo-guanidine is quite high as compared to thymine glycol. That is why these are the two important biological parameters for the DNA damage: thymine glycol and 8-oxo-guanidine. Again, there were a group who suggested they understand the effect of denaturation and addition of polyamine on the quantum yield of formation of modified bases upon the exposure of DNA to UV lasers. And what they have understand that the 8-oxo-guanidine and thymine glycols, the quantum yield values are not much affected when the denatured or spermidine effect on these the quantum yield values of these thymine glycol and 8-oxo-guanidine. So therefore, we see that they are not much changing. They are very much there. So, and the controlling factors are very less. So they are the most one who is most sensible and can be used as a biomarker for DNA damage. Again, there was a group who have understand the quantum yield values with the addition of TRISP on the quantum yield formation of oxidized base upon the exposure of DNA to UV laser pulses. And what they have understand, the 8-oxo-guanidine is quite high, is not affecting much as compared to thymine glycol and other kind of damage marker. That is why the 8-oxo-guanidine is the most recommended biological marker for biological damage in different disease progression, which is renal kidney failures and liver failures. Okay, I'm not just measurement part. I'm just going to what time we can again we can understand the DNA fragment. We can go and we can measure the whole DNA or the intact cell. So HPLC with MS detectors are the one which we normally use, and that is the most used during the quantification of 8-oxo-guanidine in the sample. During these chromatographic assays, DNA detection is the most critical steps, and if somebody have no sufficient knowledge, then it is difficult for him to understand the basic aspect. So various methods have been proposed with and without the phenols, and the background level is within the range of up to 7 8-oxo guanidine residue per 10 6 base pairs. So that is we can achieve maximum with these protocols. These protocols are easily available on Google. So if anybody would like to go, he can download the 8-oxo-guanidine quantification using HPLC, and then you can. And these are the protocol which are harmoniously uniformed and harmonized among the different laboratories in the Europe. Okay. So let us just go on the structure part. I will just take you. Yeah. So what happened that in the HPC column, we inject our sample. Then the it allows the separate the different sample on the column, and then we give a sample to UV detection. Then do UV detection. After UV detection, we can also allow the MS settings, and we can prepare a map for it, or in the form of figures. This is sort of like a peaks we observe. So around 4.15 minutes, we have a P for thymine glycol. Then again for uracil, we have a 13.42. Then for 8-oxoguanidine, it is 29.62 to 2 minutes. That is where the you can expect a P. So what we do, we normally run the first the standards, and we ascertain the peaks. After the standards, we run the sample of the blood or fluid, whatever the case, and then we try to analyze the peak area. The peak area gives the quantity, and the peak position is indicating the quality. The qualitative detection. Okay, that is how this is the most important technique we can understand. So for the different, we have a different time, so we can measure. And what we can see that again the timing glycol and quantity is the most important one. Then comet assay, another important technique where we take a cell, different cell, and we fix in the agarose gel, then through the microscope on the microscopic slide, and we can either perform the alkaline or the neutral, depending upon what we want to ascertain, whether we want to go for single stand breaks or double stand break. So I'm not going to the nitty and gritty of that particular assay. So this assay, on this assay slide, this cell is something DNA looks something like this. If you have seen some comet, it means that the DNA is damaging. If you won't see any kind of comet, it means that DNA is intact, or DNA is intact. So there is no much damage has been happened. So these are two typical figures I have been showing for untreated cell and the irradiated cell. So in the irradiated cells, you can see a comet. It means that, and because of this, it is called comet assay. So it is a DMA is got compromised. So this is, I'm not going to nitty and gritty because this is the lecture which is just to make you aware about the radiation chemistry and the DNA images. So if you wish, then you can download our papers, you can go through it, and you will get information about it. I hope so. I'm not going to much of details, and this the figures, the comet assay associated with DNA glycosases can take place, and this look like, and we can quantify the single stand breaks and double stand breaks. So this is all what we like to say in this lecture. I hope you will like it. You will aware about that how the anything radiation affect the micromolecules and how they cause damage. And we hope so that you will give your love and appreciation to this lecture series. And again, I'm very thankful to Ramanujan College for giving me an excellent opportunity to interact the audience. Thank you so much.